Our next speaker is our local gal, Dr. Tracy Matsumoto, born and raised in Hilo, Hawaii. Tracy is the research leader here at PBARC of the Tropical Plant Genetic Resources and Disease Research Unit. She also serves as our acting curator for our tropical germplasm repository. And then, of course, in her spare time, she is a scientist doing incredible work in a variety of crops. But what she's going to share with you today is specifically the work she's been doing on the transformation of anthuriums and orchids. Yeah, okay.
Yeah.
And so, welcome, Tracy.
Okay, thank you. Okay. So thank you, everyone, and it's nice to see a lot of new faces in the audience, and so welcome. So just to give you a brief overview, so actually for the orchid transformation, these were actually done at the University of Hawaii at Manoa. But these are, we did get an MTA from Tessie to bring it in, and right now we're in the process of working out logistics with Mike Shintaku to actually do the virus testing on these. So these should be coming by next year. You should have an update on how they're doing. So, okay. So I don't know, most of you, or some of you, I guess, have never seen, but this is our pipeline, and so this is our Anthurium transformation pipeline. And so this is kind of a review, but basically, the nice part about it is I just do coordinating part. So I guess that's my new job, right? Become more of a coordinator, but I get to sneak some research in there. So basically what we have is we have the tissue cultures coming through the line. So currently we're working on Marion Seaford as well as Midori. And what it does is it starts out here. We are first doing bacterial resistance. So we put in the bacterial resistance genes, in which Lisa will be the person screening those. Then nematode resistance would be Roxana, and then we have our new traits with Dr. John Suzuki, and then we come out and we're kind of in charge of the multiplication and the testing part. Oh. Okay, and so before, I guess, when we first started the project, then this is an old joke, but Dr. G was the one. If you notice, that's the pressure valve. But since he's retired, now we can have Eric doing that part. And so, no. But, okay, so that's kind of the overview of what we're doing. Yeah. So basically, I'm just want to do is give everybody a kind of an overview of what we're doing. And so just to give you, so we're going to go back to Biology 101. And so this is our plant transformation. And so what we're doing is we're taking genes for a desired trait. We have a plant regeneration system, which is the tissue culture system. And we need a method to deliver the gene into the trait. And so just to overview, we always have a gene is made of a promoter, your actual coding sequence, and your terminator. And so that's a gene that's found in you, me, plants, anything. Okay? And so what we were able to do is we have a wonderful network of scientists in ARS. So we work with Dr. Roger Thilmony at ARS Albany. And what he was able to do was to provide us with many monocot genes. And so, as you all know, corn, anthuriums, orchids, they're all part of the monocot family, as opposed to the dicot family, which is like your tobacco or beans. Yeah. Okay, and so promoters determine your gene expression. So how much, if a promoter will tell you, well, let's turn this on in the eyes, or let's turn this on, in the spadix, let's turn this on in the spade. And so those are the different things. So as they say, here's the little cartoon, and there they are. There they go expressing themselves again. But, okay. And so before we had mentioned, we also need a method of getting the gene into the plant material. And so this is kind of the this is the cowboy way of doing it, where you just pretty much just shoot it in. So what you do is you have a gas with a rupture disk, and when it reaches a certain pressure, you'll have your DNA sitting on little gold particles. And basically what you're going to do is you're just going to pop it in there. And so this is a good way to basically see, would this gene work or not before you do the rest of the work. And so what we did before is we had tested the different types of genes. So the first column you see is the 35S promoter, which is actually a promoter that's usually used in monocot genes. And so the intensity of the blue will tell you how well the gene is working. So the bluer it is, the better it would be, it's better expressing. And so you see a rice actin gene, kind of blue. Cytochrome C, you can kind of see it right here. Then we move to the maize ubiquitin. You see a lot darker blue, rice ubiquitin, and then there's a rice ubiquitin monomer that you see the much brighter blue. So in order to get our genes to be more effective in the plants, you kind of want that higher expression. And so from these, we selected the rice ubiquitin gene. Okay. And so then so that's just kind of a transient assay. So that was the leaf, but we also have to look at different tissues because as Roxana will tell you, she doesn't really care about the leaves. She cares about the roots. And so in order for her nematode genes to work, it has to be expressed in the roots. And so these are some of the things that were bombarded into root systems. And so we do get some nice expression in the roots. Okay, and just refresher, so this is just only temporary, right? We're just bombing it in, seeing if it turns blue or not. Okay, so to get a more permanent type of transformation, what we're doing is we use something called Agrobacterium. And Agrobacterium is a naturally occurring soil bacteria. I don't know if you see it. Sometimes when you see those little galls on your tomato plant or something, then that's pretty much, they call it nature's genetic engineer, because what it's doing is it's putting its genes into the plant to produce food for itself. And so what we do is we take out the genes that are for producing its own food, and we're replacing it with the genes that we want. Okay, and so basically these are the plasmids that we transform into anthuriums. So these are the food genes replaced by our genes. And so if you notice, they're driven by that same promoter. And so here we have some stable expression. So these are plants that we transformed in culture, and then we stained it, and then you can see a much darker blue color. So it looks like it works fairly well. Okay, and so what is the gene that we use? And so this is a anti-microbial peptide. So what it does is it expresses this protein, and basically the protein will just stick holes into the bacteria to kind of kill it. And so we know that it's worked before in transgenic potatoes, and so this is from a work that we actually got from another ARS scientist, Dr. William Belknap. Okay, and so what we did is we transformed these, and then we tested, are the genes there? And yes, they are. So basically then with the first few plants, what they did is actually, in fact, Braden did a first couple testing with some of these. And basically you inoculated with bacteria in culture, and then what we did is grind up the new leaves over time. And what you can see here is you can see the untransformed control. So the yellow thing that you see up there is the bacteria, but you can see the ones that were transformed, you don't see the bacteria really reproducing from those lines. Okay. But does it work in the greenhouse, right? Because that's the next question. So here you can see the bacterial resistance gene. So we verified that the gene of interest is still there. And then so in this first column, what you see is the control plant. So this is the untransformed plant. And then what Donna did is she notched the edges with bacteria on it so that it should die over time And so here you see our two transformed lines. And so you can see that they are, they seem to be doing well and not dying like the control lines. And so those were the two surviving plants. And so Donna did another test because Donna's our best critic. And she's like, okay, I'm going to really make sure. So she tests them again. And then you can see that the plants are still alive. So it's been, I guess, over a year. And so then what we also have is these are non-transformed Marin Seaford. So these are different lines as well. And you can see the, so these are the non-transformed lines with water-only control, I'm sorry, water-only control here, as well as the bacteria infested. So, and then what you can see is over time, the water-only control looks good, and the other ones succumb to the bacterial blight. And then we have our transformed lines here, water-only control, and they look a lot healthier. So, oh, I'm sorry. Okay. And so right now, what we're in progress, because in order to convince Lisa that these do work, because sometimes potted plants react a little bit differently in pots versus field trials. So while we're in the process of getting our APHIS permit, what we're going to do is test them. So Lionel was nice enough to give us space in his greenhouse, and we're testing them in beds. And so these, you can see them later on today. But just a word of caution, if you're going to go on to Greenpoint Nursery, I wouldn't necessarily go into this greenhouse. We'll have you stick your feet in 20% bleach, but I don't know, if they're going to go on, then maybe we want to skip this part. But if you're not going to go on to Greenpoint, then, or not going to go on to your own farm, then you're welcome to see these. Yeah, we don't want to be the cause of everyone's bacterial blight. So. Okay. And so the next steps, what we have is, so we know that we grew some plants in the greenhouse that have the GUS, and then because is the expression maintained once we get it to the greenhouse. And here it's staining, and it's staining really well. And here you can see it's staining throughout the whole root. So now Roxana's happy. And so basically now what we can do is we do have a construct that was made by Roger. And so these, right now we have the single construct that we will transform for nematode resistance. And he's also working on the double construct, which will contain the bacterial blight resistance as well as the nematode resistance gene. And so this is the first gene that we're trying for nematode resistance. And I'm not sure if Roxana is going to tell you about the new ones, the potential new ones coming up later. So this is, like I said before, this is all a group project. We have to thank all of our cooperators. I guess I don't have a picture of Braden and Lionel, but Donna does all of most of the work here. So just to let you know. And with that, any questions?
How soon before we get it to trial where we can start growing it out?
Probably. So right now, as we're trying it out in there, Donna's actually multiplying to get bigger scale, and then our APHIS permit. We're working on the APHIS permit. Yeah. So hopefully it should be.
In a year.
Yeah. Hopefully in the next few months to try it in the field. Correct.
And the field test is just for bacteria?
Right now it's just for bacteria. Yeah. But yeah, the reason why we start with bacteria is that sometimes Roxana will lose the plants that due to bacterial blight, so that she can't evaluate it. So we're starting with bacteria first, and then adding on the nematode there.